Lift Events and Experiences, known for Canada's #1 cannabis conferences and trade shows, is hosting a full slate of events in 2023 and expanding to the U.S., and you're invited. Lift welcomes everyone from legacy participants to multi-state operators to everyday folks who enjoy and respect the plant. Join us in lifting the cannabis community together, January 12th through 14th in Vancouver, then Toronto in June, and San Francisco in August. Visit www.liftevents.com for details. That's www.liftevents.com. Welcome to the Cannabis Cultivation and Science Podcast. I'm your host, Tad Hussey of Kiss Organics. This is the podcast where we discuss the cutting edge of growing from a science-based perspective and draw on top experts from around the industry to share their wisdom and knowledge. Before starting the podcast, I did want to address one major change to the show. I know I mentioned in the past about adding sponsorships or ads to the podcast. I've avoided it to date, but with rising costs and changes to the economy, it's now something I've had to revisit. Basically, moving forward, there'll be 1 to 2 short ads now at the beginning of the podcast. We will also be adding the Patreon again with a subscription-based model where for $5 a month, you can get a link to commercial-free version as well as other perks. Please reach out directly if you have any questions, and I guarantee the podcast content will always be free and accessible. Anyway, back to today's show. Our guest today is Jeff Lowenfels. I wanted to get Jeff, our very first podcast guest and old friend, back on the show. Jeff is the author of the popular teaming series, Teaming with Microbes, Teaming with Nutrients, Teaming with Fungi, and now his new book, Teaming with Bacteria, which he joins us today to discuss. He also has a book called DIY Autoflowering Cannabis, which we discussed in an earlier interview. Jeff is an extremely well-respected and popular national garden writer. He is the former president of the Garden Writers of America. Jeff has been a good friend now for over a decade and is a wonderful advocate for organic gardening, compost teas, and the microbes in our soil. Thanks for coming on the show, Jeff.
Hey, I am excited. You know, I have a relationship with you and your family, and it's always a pleasure to be here.
Awesome. I really appreciate it. I always love talking to you. I think this is your 3rd time now on the podcast. You're one of my most frequented guests.
Yeah, I think it is.
And I'm really excited to talk about your new book and some of the new research you've come across. Why don't you take it away?
Yeah, sure. Well, you and I were steeped in the soil food web, and I wrote a book about that called Teaming with Microbes, and that book came out in 2006. And essentially, it was a codification of what we all learned from Dr. Elaine Ingham, who's, of course, the guru of the soil food web. And basically, the system, as everybody probably knows, operates off of the exudates that are produced with photosynthetic energy by the plant. The plant designs those exudates, drips them out into the rhizosphere, and they attract bacteria and fungi that need the carbon that are in those exudates. Remember, Elaine called them cake and cookies, sugars, and but there's a lot of carbon in them, and that's what these photosynthesizing organisms need. And so they're very happy there in the rhizosphere, but they are attractant to their own prey, or things that eat them, and those are nematodes and protozoa. And so the nematodes and protozoa eat the bacteria and eat the fungi, and as a result, there are they also need carbon. You and I need carbon, of course. Everybody operates off of carbon, but they don't need everything that's in the bacteria or in the fungi. And so that excess is plant nutrient, and it's in plant usable form. It has a charge on it. The microbes essentially put a charge on these excess nutrients, I guess we'll call it that, and they're able to move from the soil into the plant. And of course, Teaming with Nutrients is a book I wrote that talks about how that happens and how they get in the plant and what happens inside the plant. It's kind of a fit together. And so that was the soil food web. I think at that time in 2006, I had a paragraph on mycorrhizal fungi. And it wasn't until 2000, I revised the book in 2011 to add a chapter about mycorrhizal fungi, which are these specialized fungi that are attracted to the plant by the plant exudates. The plant signals the mycorrhizal fungi that it's okay to enter the plant, and it does enter the plant in the root system, in the meristem area, and the fungi enters in between the root cells, and there's a transfer of exudates and goodies from the soil by the fungi, and so this was a very important addition to the soil food web. Well, it turns out that in 2010, a group in Australia, and unfortunately, and I really just got to learn how to pronounce this woman's name, a wonderful woman who has a hyphenated name, Lonheim, and her team in Australia discovered that some of these bacteria that are attracted by those exudates to that rhizosphere area actually enter into the meristem cells. Now, this had never really been seen before, or if it had been seen before, the implications of what it was not fully understood. And so this group studied this, and they termed it, I'm going to give it the American pronunciation, rhizophagy, R-H-I-Z-O, rhizo, of course, meaning root, and phagy meaning eating, so root eating. And it appeared they weren't quite positive what was going on, that perhaps the cell was opening up, engulfing bacteria, and then closing up again. And so that was why they were inside there. They didn't really study about what was going on, and apparently their grant funding must have run out. And so it languished. Now, I remember getting a note from one of my friends who read about this, and I read the note, and it just said rhizophagy. I had no idea what it was until I ran into a gentleman who's a professor at Rutgers University named Dr. James White. And what Dr. James White and his students were able to do, they could trace what was going on with those bacteria, how they got in, what was happening with them. And what they discovered was beyond astounding and changes the picture of the soil food web so that if you don't understand rhizophagy, you're not going to have the full picture of how to operate in a soil food web situation. In other words, how to grow your plants properly so that you are teaming with the microbes and the soil food web is helping you. And so I think what I probably should do, with your permission, is describe the rhizophagy cycle. Does that make sense?
Yeah, that sounds great. Why don't we start there?
Oh, okay, we will do so. I wish we were doing this, with a super screen in front of us, because the photographs of this cycle are absolutely phenomenal. And in the book, I've got a lot of them. You should be able to find some of these on the web. Before I begin, I think I want to point out that bacteria are something that we don't study. They're too small to really see. They're 90 times smaller than a hair. They're just tiny, but they're everywhere. And so as a consequence, we really, when I say the word bacteria, people think of, like a car in a driveway. But when I say that's a bacterium, when I say bacteria, you should be thinking of the New York City's biggest airport parking lot, and all of those cars are bacteriums that form bacteria, and they're everywhere. And you should immediately, after listening to this podcast, Google bacteria head of a pin, and you'll see some unbelievable pictures. And just last week, there was an award given to a picture of and you can Google this too, a single tongue cell surrounded by bacteria. And you'll get a feeling for the tininess and the multitude of these organisms that multiply every 20 minutes on average. They are just phenomenal, and a part of the book deals with bacteria, obviously, and describes them and how they operate and all that kind of good stuff. All right, anyway, so now we've got these exudate-attracted fungi and bacteria that are up there in the rhizosphere area, And some of these bacteria, instead of being eaten, they smell a popcorn smell, a buttered popcorn smell. It's butyric acid. It's the same stuff that's in your stomach, and if you get too much butyric acid together, it smells rancid, and if you get a tremendous amount of butyric acid together, it smells like vomit. But at this stage, down at the rhizosphere level, they're smelling buttered popcorn, in my imagination, and it causes them, because they're living in a little bacterial slime, to think that there's an area outside of their home slime that they can move to where they'll have a better opportunity to survive, to eat, to multiply without having to deal with all the other people in the slime or other bacteria in the slime. So they push against the smell, and it turns out that what they're doing is moving through the root meristem cell walls. The root meristem cells they're brand new. They have very thin cell walls. They're very immature cells, and they are invaded by these bacteria who enter into a space between the cell wall and the membrane that holds the cytoplasm in the cell. The cytoplasm, the place where all the organelles are held, the nucleus, the chloroplast, blah, blah, blah. The way I liken this is to a tofu container. So you have a white tofu container. That's the cell wall of the plant, the meristem cell wall, very thin. And the bacteria go through there. They're very small, so they go through there, and they end up in that watery area that's in a tofu packet. That's called the periplasmic space. And then you've got the tofu. Some of them actually enter into the tofu. That's the cytoplasm membrane representation. So they're in this periplasmic space, and the plant goes, I just got invaded by bacteria. Even though I invited them in, I need to make sure that they're not going to harm me. And so the plant sprays the bacteria with a superoxide, which is very strong and has the result of stripping off the cell walls of the bacteria. So these are now what we call L forms. They have no cell walls, and the material that was in their cell walls is absorbed by the plant, contains micronutrients, metals, some really neat things, and the plant uses them as plant nutrients. Internally, they're internal and they're absorbed internally, and they're used internally, obviously. Then the bacteria goes, whoa, wait a minute, we don't like this. And so the bacteria produces an antioxidant to reduce the superoxide's ability to destroy the cell walls of the bacteria. And this turns out to be nitrite. Nitrite is then converted as a result of a series of chemical reactions in the periplasmic space, some of which involve the plant, into nitrate. And the nitrate is plant usable, and it's absorbed by the plant as well. Up to 30% of the nitrogen that a plant can receive can be through this cycle. Now, if that wasn't enough, it turns out that these little guys, they're breeding and they're multiplying every 20 minutes. And as they multiply, they're also doing something else. They're producing a phytohormone, a plant hormone that we call ethylene. And ethylene causes the meristem cell to grow. So here you have these invading bacteria. They no longer have a cell wall. They're multiplying. And as they multiply, they form these funny little balls. They look like beach balls, volleyballs, soccer balls maybe. They have 4, maybe 6 wall-less bacteria stuck together. And they're circulated around this periplasmic space, producing ethylene, which is also circulated, causing the meristem cell to grow. Then they get so plentiful that they end up getting trapped against the cell wall of the plant, the meristem cell wall, where the ethylene is produced in a concentrated area because it doesn't get cycled. And that ethylene causes the meristem cell wall to form a tube. You and I know this tube to be a hair, a root hair. But this tube actually begins to fill up with these bacteria. And as they get more and more in, the root grows, you're getting more ethylene production, and the root hair grows, and it grows longer. The bacteria are literally expelled by a periplasmic liquid tidal wave out of the root hair tip into the soil, where they go back and they regrow their cell walls, and eventually 2 or 3 days later go back into a new meristem cell and repeat the trip. And they're not just blown out once. Those, as that root hair grows, there can be 4 or 5 of these explosions of bacteria that end up back into the soil, some of which obviously end up getting eaten, and they're part of what I like to call the soil mediated system, where they travel into the, but some of them go back and do the trip again. It's just simply incredible. So what we've got essentially, and I'm going to refine this just a little bit more in a minute or two, what we've got is the Dr. Lane model that you and I, breastfed on, if I can use that term. That model is a farming model. The plant's a farmer. It's throwing out the exudates. It's helping it's fertilizing the bacteria and the protozoa and everything. It's all, and then they end up, taking in the resultant crop. Mm-hmm. What we've got in this rhizophagy cycle is a ranching situation, where the sheep are brought into the barn, they're sheared. We use the wool. We eat a couple of lamb chops because what the heck, they're there. And then we put them out in the pasture again to regrow their wool. And then the following season, we take them back in again, and we take the wool off the sheep again. So we've got a ranching situation and we've got a farming situation, both of which are part of the soil food web. And so now we've got a much more complete picture of how that plant gets its nutrients. If it's getting 30 or 40% of its nutrients from these endophytic bacteria, endophytes meaning that they live inside the plant for part of their lives, then this is a major, major new discovery. And I say it makes teaming with bacteria equally as important as the original book, Teaming with Microbes, because this is such an important thing that's going on. And the implications of it, both in the future and for right now, are stunning and usable. So we'll certainly, I'm sure, be talking about that. Any questions about me so far, my stuff so far?
Well, so I've read the book.
Yeah.
Listening to your explanation right now is actually really helpful. So if you're someone that has read the book and then listens to this, I think that's actually not a bad direction to go, because you break it down really well, your explanation. I love that analogy. So I'm going to try and put it into my own words to simplify it even further, and please correct me where I screw it up, because I'm still learning and processing all of this myself. So basically we have a cycle where these bacteria, specific bacteria, are going into the meristematic tissue, or the meristem, which is all the new undifferentiated tissue that most people are familiar with, the apical meristem, where you have, all of our new growth is based around meristematic tissue. So it's like really the exciting place on plants: new root tips, new leaves, and very thin walled, because they're brand new cells. So so they become more specialized as the plant needs them. But they just start as really like a blank canvas of a cell, is how I've always thought of them. So it's kind of where all the action's happening. And now you're adding in this thing we didn't even know about, which is that there's all these bacteria that work their way in, lose their cell wall, or their wall, what you referred to there, while they're inside. They have a cell wall. They do have a cell wall. Okay, they lose that wall, which is pretty amazing. And I have trouble visualizing this, but then as they complete that cycle, then they're going back out, regaining that wall, and then starting the process over again. And then Well, I just wanted to ask, so then you mentioned nitrogen. Sorry. You mentioned nitrogen as being the primary nutrient in this, and specifically nitrite converting to nitrates. Are there other nutrients that we know about that are happening in this equation?
Absolutely. I mean, depending on whether it's a gram-positive or a gram-negative cell wall, the material that makes those cell walls is absorbed, and so I don't have the list in front of me, but it's an interesting list of things. They are amino acids and other things, and so there's quite a few nutrients that are available by the rhizophagy cycle, which means, I guess there's really sort of two questions. I'll let you continue, but why would a bacteria do this, and why would a plant let the bacteria do this? I mean, the bacteria is an enemy of the plant in many instances, and the plant knows that and has defensive mechanisms to keep bacteria out. That RO in particular can be strengthened. And incidentally, when it sprays that RO, it has the capability of removing its own cell wall, which is very thin, and so it has to strengthen itself in order to be able to handle the rhizophagy cycle, and that strengthening lasts throughout the life of the plant. So again, the implications are just mind-boggling. But yeah, there are a series, I don't know, 6, 7, 8 nutrients that actually get into the plant as a result of the rhizophagy cycle.
And there's a bunch of different strains of endophytic bacteria. This isn't just one particular one. There's quite a few.
Yeah, and that's a very important point to make. If I can just spend 30 seconds and explain that there are other kinds of endophytic bacteria that enter the plant through branching in the root system as it branches. There are little cracks in there, and they sneak in. They come in through stomata. They sneak in through wounds and things like growth holes and things like that. And so they're in the plant. Now, with regard to cannabis, this is just too stunning and too amazing. What some of the studies have shown is that the mix in the soil of bacteria that become endophytic or are capable of becoming endophytic bacteria, either going through the rhizophagy cycle or going into the plant to do other stuff, it's always basically the same. But once you get inside the plant and categorize the endophytic bacteria that are in that plant, they differ from strain to strain. Holy crow. This is the, quote, terroir that we have been looking for. It's the bacteria. There's, let's pick a strain, Girl Scout cookies has one kind of endophytic bacteria that's different from, naming another strain, it's just incredible. So now we're going to be able to start doing some research and develop strains in different kinds of ways. This is really big news for cannabis growers. And these studies will become more and more plentiful as President Biden's initiative seeks a home. We're almost there. We've legalized cannabis where we can do the research that we need to be doing in order to get the very best of best. So, it's just incredible. Now, these endophytes, when they go in the plant, produce phytohormones. So they've got the capability of producing auxins and doing everything that an auxin does, because they're real auxins, or gibberellic acid. Ethylene is a phytohormone. You've got about 6 or 7 phytohormones that are produced by these bacteria, and they help the plant do all sorts of things. And so the other reason why a plant might let an endophytic bacteria survive inside itself is because the plant is getting helped by that endophytic bacteria. That they're using those phytohormones. Again, simply a stunning implication when you consider that all of what I call, let's just call them the big bad agro guys, what are they studying right now? They're studying the capability of bacteria, endophytic bacteria, and some fungi, to produce nitrogen in the plant. That's what we have in the rhizophagy cycle. That's nitrogen fixation, just like in rhizobia, although it's not quite the same system, but it's internal nitrogen fixation. Holy crow, isn't this what we need? A wheat plant that's capable of producing its own nitrogen by hosting the right kinds of bacteria. Yes, and that's what that's the holy grail of agriculture these days, and we're getting there. And part of it is because of the rhizophagy cycle, and part of it is because of this endophytic phenomena that we're stumbling on and we're beginning to figure out how to use.
So where is the bacteria getting the nitrogen in the first place?
It's pulling it out of the air.
So, but not all of these are necessarily rhizobacteria, correct?
That's correct. That's absolutely correct. And that's one of the stunning things about this. So in a rhizobia situation, what you've got is a bacteria that's attracted literally to a root hair. How interesting. The root hair curls around the bacteria. These bacteria convert themselves into bacterioids. It's a little bit of similarity, doesn't it? It's sort of interesting. So it finds itself inside the plant in a different format. And if there's a system whereby the oxygen level can be reduced, they are capable of producing nitrogen from the air, and they use that nitrogen to live. It resides in that nodule. A little bit of it leaks out into the soil. When the plant dies, that nitrogen is released back into the soil. So in this situation, we've got these endophytic bacteria inside the plant producing nitrogen just from the air. Now, with regard to cannabis, it becomes even more interesting. That paper I sent you this morning by Dr. James White blew my socks off because it proposes a system whereby there's an explanation for what's going on inside glandular trichomes. And if there's anything that a cannabis grower is interested in, it's glandular trichomes. And so what the theory is now, and incidentally, in the book, I alluded to a lot of this stuff, but the book was written 3 years ago, believe it or not. It goes through the editing process and then the printing process, blah, blah, blah. So it takes a while to get a book out. So at the time, these papers had not been completed. And so I was theorizing what they would show. But now Dr. White and his students have demonstrated enough to come up with a pretty solid theory that there is bacteria in these trichomes, because originally people were beginning to think that these trichomes look like root hairs. Why isn't, is there the same kind of nitrogen fixation activity in them that there is in root hairs? It turns out there's not quite as much, but there is nitrogen fixation going on. The bacteria that are in these trichomes turn out to have to be in a situation where there is a oxygen-deprived situation in order to fix the nitrogen, and what deprives them of oxygen is being bombarded by, are you ready for this, cannabinoids. Holy crow. Being bombarded onto these bacteria so that they end up producing nitrogen in the trichomes, but what gets produced as a byproduct is what people grow cannabis for. Wow. So when I say this is an important book in terms of its amendment to Dr. Elaine's Soil Food Web, this is also an amendment to anything you've been thinking about with regard to cannabis. And it goes even further, because the way these bacteria survive from generation of plant to generation of plant is through the seeds. They get into the seed situation. They get into the flower. They get caught up when the flower forms the turgid around the embryo situation, and they're inside. Some of them right up by the embryo. Some of them probably in the embryo. Dr. White points out bunches of them in the seed coat itself, and a bunches of them just inside the seed coat. And these are the bacteria that jump out into the soil once you germinate the plant or the seed and start the process of providing the right rhizophagy and endophytic bacteria for the plant. Holy crow. All these years, so many cannabis growers have been sterilizing their seeds. Whoa. They haven't been fortunately able to fully sterilize their seeds because it's not that easy to do. But they've been taking some of the important bacteria out of the system and then, probably wondering why they're getting powdery mildew, et cetera, because these bacteria do all the things that Dr. Elaine explained. They produce antibiotics. They have, all sorts of things. They go out and create iron and minerals in the soil and metals and bring them into the plant. And we plant things in paper towels. How about that paper towel germination process? It's in my cannabis book, the Autoflowering Cannabis. Yeah, well, you know what? You better be planting that paper towel piece in the soil as well, because you've got bacteria that jumped off the seed onto the paper towel. You want that in the soil. All the advice that you've gotten over the years about not throwing, from me, as a result of what I'm gleaning from Dr. Elaine, not throwing away soil unless you've got a terrible root problem, because that soil contains exudates. Well, it also contains the bacteria that the cannabis needs, and it contains probably the right bacteria from previous grows that are either dormant or in there waiting for you to give them exudate so that they can help that plant become the best cannabis plant it possibly can. This is important stuff, and it goes one step further. And then I'll stop for a second and take a deep breath in. There are bacteria that people are now capable of buying, and that some of the grocery stores are beginning to carry. I'm so terrible at pronouncing these. Azospirillum, for example. There are a couple of bacillus that people are using. We already use cover crops. It's kind of interesting. And the reason we do that is because they fix nitrogen and put the nitrogen in the soil. So all of this ties together. God bless Dr. Elaine and God bless, Dr. White, because they really are geniuses for codifying all of this stuff. None of this stuff is stuff I invented. All I do is report it. So.
That's a lot. I have a lot of questions. I don't know where to start. The first thing that comes to mind for me, and it's a little bit, it goes back a little bit to what you're talking about earlier, though, is so inside of the plant, we have all these endophytes. And like you mentioned, it's
right.
Endophytes and endosymbiont, I guess, something that lives within the plant that's beneficial. They're both benefiting from the relationship. And that
that's
could
correct.
be a bacteria, it could be fungi, it could also be archaea.
Could be a virus.
Yeah, micro-eukaryote. There's all these things inside that are going on. We're not existing in a vacuum is essentially what you're getting at.
Right.
How much of what you're talking about applies to archaea as well as bacteria? Are you differentiating between the two, or do they just have very similar functions to where it's...
It's an excellent question. I don't know. And I will try to find out. I've never seen any references to archaea in this regard, but, how do you separate the two? I don't know. It's a very interesting question. It seems to me that they clearly have a cell wall, even though it's a little different than the cell wall of a bacteria, that would contain nutrients that would be useful to the plant. I don't think they produce nitrogen fixation. I don't think they're capable of nitrogen fixing. They are part of the process of nitrogen fixation in soils elsewhere. So yeah, it's an interesting question. I'm going to have to take a look at it, and we'll have to do a whole nother show on it.
It might be another book, huh? Yeah.
God, another book.
You say that every time, and then you always end up getting excited and writing another book. So I feel like it's coming.
Only when there's something that comes forward that that adds to this thing that Dr. Elaine infected us with. Once you become a soil food webber, you're stuck because you can't go back. You can't deny the science that exists out there. you can only improve on it, and that's what happens. And so we've become better and better at this. It's really something, really something.
Yeah, I mean, I started with you and Dr. Ingham in this journey through meeting you both through my father, as you know, decades ago. And that's really where I began. And this whole idea, these were your words, like the plant's putting out all these exudates, the plant controls it. You had a wonderful graphic that went with it, and you'd have a picture of a bush. And you're like, if the plant wants Japanese food, it would select, for the exudates for that. And then you had a picture of some Japanese people would show up in the photos. It was very entertaining, but it really drove that idea home. And so it's more what you're saying now is we still have that process going on. We still have the nutrient cycling, that microbial loop
absolutely
going on. But on top of it, we also have these endophytes, the fungi, the bacteria, the archaea, circling inside the plant, infecting into the plant in also a symbiotic way.
Right. Yeah. I mean, I think the way I look at it is that there's three basic ways that a plant takes its nutrient in via the soil food web system. And the first way is this soil mediated movement. The stuff is dumped into the soil and it finds its way into the plant. The second way is the mycorrhizal situation where the mycorrhizal fungi takes the nutrients that are in the soil, sometimes actually converts, and then brings them into the plant. And then the third way is this rhizophagy cycle. There may be fourth and fifth and sixth ways. I don't know. But so far there are these three ways. They are all based on the 2006 model, which is you've got this pool of bacteria and fungi that are attracted to the plant by the exudates, so they fit. There could be other stuff. I don't know what it would be.
Well, there's also just straight osmosis where the plant will just take in.
Well, that's the plant. That's the plant mediated system.
Yeah. Okay, that makes sense.
Yeah. I mean the soil mediated system. I'm sorry. Yeah. And I don't even know if that's the right word. You got to come up with these things, figure it all out. It's such new stuff. But again, this is really important stuff for people to understand. It doesn't change the soil food. You still don't rototill. Now that when I used to say the bacteria end up, that are in the root zone end up at the top or in the bottom, and they're all displaced. Well, that is the same thing whether they're rhizophagy bacteria or whether they're just rhizosphere bacteria. So you don't rototill. Compost tea might have a more ethical, more understandable reason for working in some instances and perhaps not in others. The addition of the right kind of mulch in order to make sure that you're fostering good bacteria is still in play. So nothing really changes in the soil food web other than the potential of a future where we can tweak the soil food web in ways that we're not capable of doing now, and certainly we're not capable of doing in 2006 when we barely even, I don't think in 2006 there were commercial mycorrhizal fungi. Now there are. That's a heck of a change.
Yeah, I mean, I think we have a lot still to learn.
Yeah. Oh, God, yes.
I think there's so much we don't understand about what's going on directly under our feet in that plant-soil, plant-microbe interaction.
Right.
I heard a soil scientist years ago mention that, he goes, we know more about what's going on at the bottom of the ocean and on the moon than we do, directly under our feet. And
an inch under our feet.
It's just it's so complex. And so this is where I may differ a little bit in this because we have some camps here around, soil food web, and you have people that believe that, it's all about the microbes. And so as long as we're testing the microbes and, taking care of the microbes, we're not going to have to deal with disease or fertility or any of those issues. All of those things are symptoms of microbial problems. And that's kind of where I would put the soil food web camp. Now, on the other end, you have sort of the mineral guys, the Albrecht, Solomon folks, where they're like, let's focus on minerals, get the minerals balanced into a form that the plants are going to have optimal fertility, and then all those microbes are just going to show up. I'm kind of like right in the middle there. I don't think either of them
me
totally agree. Like, I want to add the minerals, but then all my behaviors are really towards taking care of these microbes in this process, like you mentioned. Limiting tilling, reusing soils where we can, adding organic matter in the form of compost. All of these things. Right.
Yeah, well, and I think, Albrecht makes tremendous sense, and obviously it's tied to the microbes. Because plants need those microbes to grow, and we know what happens when you take the microbes. For example, in the book, I discuss this, and Dr. White's shown this a number of times. If you don't have the bacteria, you don't get root hairs. Whoa. And the roots don't grow properly. Whoa. And then having taken the bacteria out, if you put the bacteria back, you grow root hairs. Ooh, that's pretty interesting. But what is a mineral? A mineral is a microbe food. Sure. In the right situation. Now, when Albrecht breaks down is when people don't do it properly and the ratios aren't proper. That's sort of like applying too much chemical fertilizer. It screws up the system. The microbes can't operate. It's not that there aren't any microbes. The microbes that like the fertilizer are not necessarily the microbes that like that the plant likes. And so, I'm with you. I'm right in the middle. I think there was a big fight at one point in time because people were yelling at Dr. Elaine Ingham because she apparently or supposedly, and I'm not trying to put words in her mouth, said that if you've got a good soil food web system, you don't need to add any nutrients. Well, I think in the sense that
I think the quote was that all soils contain adequate minerals for growing healthy plants, or it was something along those lines.
Yeah. I'm not sure. I'm not sure whether I can. I'm not sure I can fully comprehend that. If that's the right way to put it. But there's no question that if you use things that replace these bacteria, what they call the nitrogen efficiency ratio goes down. So the natural system seems to be the most efficient system. Are minerals part of the natural system? Yes. Whether you go with that quote or don't, the minerals that are in the soil are helpful. And if you don't have those minerals, it doesn't hurt to put them in. It just makes sense to me. But that doesn't mean you don't need the microbes, because you do.
Absolutely. I just wanted to say, like, we know that biostimulants, and that's just sort of the industry term that I hear for adding microbes.
Right.
So any of these products that you mentioned that have bacillus or azospirillum, all of these things, mycorrhizal fungi, these all be called biostimulants.
Yes.
Is one of the fastest growing areas of sales and research in agriculture. Yes. Not even just specific to cannabis, just agriculture, which is huge, like how we grow our food.
That's why I say I call them the big evil chemical companies. They're all studying this stuff. This stuff replaces chemicals because it's natural and it works, and you don't have to continually apply. It's phenomenal, and we'll get there because it just makes so much sense. We forget. No, we don't forget, but a lot of people don't understand that in order to grow the food the way we grow it now with combines and chemicals, et cetera, we're destroying our soil system to the point where it's, the experts say we got 61, I don't know why they don't say 60, but 61 years of harvesting left of soil. Whoa, really? Well, that's because we're not using bacteria that give the soil structure, and fungi that tie those little aggregate of slime stuck together particles together, et cetera. So, yeah, everybody's moving this way, and it's a good thing, I think.
Yeah. So one of the things that we see is by adding specific bacteria, even with so much that we don't know, for example,
Right.
we see increased growth or disease resistance or whatever is associated with it. And some of these things are symbiotic. It's not even the sum of the parts equal themselves.
Right.
Sometimes
No, there's a lot of
3 + 3
Right.
= 9. We're finding with the way these microbes work together. All that being said, are all of these microbes in most soils or media? Like, would compost contain all of these for the most part?
This is an interesting, very interesting question. Great question, because you see these studies that say there are about 650 kinds of bacteria that are in all soils around the world, et cetera, et cetera. Of course, that's a teeny little number. The bacteria, I don't know how to put this. Ask the question again, so I can kind of.
So essentially what I'm getting at is, because I'd heard a statistic that like 99.98% of world soils contain the same bacteria, more or less. Like they're pretty much everywhere. Now, if that's the case, why is it? So we're essentially just giving a booster shot when we add these nutrients to give that particular strain or species a jump start or an advantage over other bacteria in the soil that may be innocuous or disease causing or less beneficial. Is that the way to approach it?
If you believe that all soils have the same bacteria, I'm not sure I believe that, because it's obvious to me that there are bacteria that we, in the experimentation and in some of these studies, they're putting specialized bacteria into different kinds of plants. Well, why do they have to do that? I mean, isn't it in the soil already? And you see where I'm coming from? I mean, they're looking. Not all bacteria will do the same thing in every plant. It's not quite the same as mycorrhizal fungi, which, for lack of a better term, tend to sleep around. We only can make about 8 to 12 mycorrhizal fungi out of the how many 350 that there are, but they're able to infect lots of different kinds of plants. One fungi can affect many different kinds of plants. Not necessarily so with the bacteria. That you've got to have the right bacteria. So what might help a strawberry plant grow better strawberries, a bacteria, might not do the same thing for a wheat plant or a apple tree. And so you've got to find the right bacteria, and it's probably more than one, obviously, to feed the plant. And so that tends to point to me that there are different bacteria around the world or in different kinds of soils. And then I think again about what my understanding of how Dr. Elaine Ingham sort of came upon a lot of this stuff. She collected soils from plants that did fabulous around the world. So if you had a banana plant that did fabulous, this soil from the fabulous, this is the ideal kind of soil you want to have for a banana plant. Well, not all banana plants grow the same around the world, do they? I mean, so I don't know whether I believe that original statement. I guess that's my problem.
Well, that's fair. I read it a long time ago. It could be a
it's worth looking into, because I hear it all the time, and I just haven't done so, and I think it just makes sense to do so.
I almost look at it like we're replacing or adding in sort of like the pro athletes of the microbial world when we add these bacillus species. And they may be out competing. Well, I think we have to be to a certain extent, because we can do research that shows when we work in sterile media, and then you add in these specific strains of bacteria, we get this level of increased plant growth that we may not see in a control, which could just be either a sterile environment or a variety of different soils.
Right. We certainly goes against, again, the original statement that the 99% they're all the same. That doesn't make any sense to me unless we're talking about concentrations. I mean, for example, the Alaska humus that we used to sell, I mean, that stuff was loaded with things. But of course, it also had different minerals too. And frankly, I tend to be more interested in the soil food web side than the mineral side. I probably should spend a lot more time. I am familiar with Albrecht, but we tend to be more soil food web, and might have a lot to do with the minerals that supports the soil food web. I don't know. But I do know that individual bacteria are required for individual plants. They don't all work on the same one.
This is why I kind of feel, though, that like they must be everywhere, because we can grow plants in a variety of different conditions, in a variety of different soil or soilless media, and still have a thriving plant without trying to select for a particular bacteria. So I think it's more about like I think of viral load, like a bacterial load. In the same sense.
I go back to that 650. I think there's probably a base that all plants have, that all soils have, and then the other stuff is extra or different. It's just give a little bit of diversity. Has to be. It has to be.
And I just want to throw this out there so people will get the wrong idea. I'm not a huge Albrecht guy. I think his stuff is really interesting. But I'm not a fast and true on the mineral balance side either, so I don't want people to walk away from this thinking that.
It would have been interesting had he had some of these microscopes that Elaine had and that Dr. White had. It's be interesting to see. I found a guy, incidentally, around the corner from where I stay when I'm in Portland, who has the only storefront electrical microscope. You can go in and get things done right there off the street. Holy crow. And I think he has a confocal microscope as well, so maybe we can see some of these rhizophagy bacteria.
Well, that would be fun. I'd come meet you in Portland to tour around that
wouldn't
stuff.
That be incredible?
Yeah.
Yeah, let's arrange that. That would be phenomenal. Absolutely phenomenal. Well, again, this book is, I think people need to understand that it changes the picture of the soil food web, but it is not in any way to denigrate, our pre-rhizophagy cycle understanding of the soil food web. It just adds to it. I don't see any detraction from it. In fact, as I was doing the research, all the stuff I call them cetaphores. I never know how to pronounce the word. These wonderful bacteria that are able to go to put out into the soil the ability to create different metals, either bring them into the plant or tie them up so that they can't get into the plant and poison the plant. The ability of some of these bacteria to work with fungi and change nitrogen into a usable form, and oh my goodness gracious. And then just the bacteria themselves. I'll never believe the 5-second rule again.
That's been proven wrong, unfortunately. I wish it was a thing.
Yeah, I know. And the pictures of them, they are the most incredibly beautiful organisms. And because of the ability now to use these various microscopy, photography, systems, holy crow. So when you look at a flagella under, some of these microscopic pictures, I mean, you would swear you're looking at the internal parts of an outboard motor. It's not, you see the diagrams of them and you go, wow, that's really cool. When you see it in working, or when some of this stuff is just incredible. There's in the back of the book, in the appendix, I list a website, which most books never do, and I had to really sort of get the publisher to allow me to do it, that shows this root hair growth and the root hair oscillating, and you can almost see the new molecules being put into the wall, the root hair as it grows. I mean, it's just the stuff that's out there. You can see individual DNA. Oh my God, there's stuff out there. So, yeah, we live in a terrific world. It's completely different than it was, and I can only imagine, what's coming up next. Only imagine. Well,
you had mentioned now that you have a new order that you would suggest that folks read your series.
Right.
Why don't you? Can you touch on what each book is just give a brief synopsis,
sure.
and then also in the order that you would suggest new readers might tackle them.
Right. Yeah, well, I would obviously start with soil. Not obviously, I would start with soil teaming with microbes. It explains the soil food web. It gives soil food web rules, but it doesn't include the rhizophagy cycle, and it doesn't include more than a paragraph on fungi. So the next book I would read would be Teaming with Bacteria, this one that I just wrote, and it describes the rhizophagy cycle. And it describes the role of endophytic bacteria, the phytohormones, what they do to plants, and doesn't have rules, but it does point out some bacteria that work better with different plants. And then, of course, there's teaming with fungi, which is about the mycorrhizal fungi and how they operate. And then I would read the last one, Teaming with Nutrients, which explains how these nutrients get through the plant membrane and what happens to them once they're inside the plant. And so those are the 4 books. They're all available on Amazon. I think Timber's thinking about putting a set together. I hope they are. And I always tell people that, again, this is not stuff I invented. I didn't write it. I'm not the genius. Don't give me the credit. This is stuff that Dr. Elaine Ingham, Dr. James White, and other really terrific scientists have put together. All I do is try to dumb it down so that I can understand it, and it turns out that some of you are a little bit more intelligent to me, so my dumbed-down version works well for you. And again, I mean, there's different things in each book about, that you want to experiment and try to find the right answer for. So for example, you asked me about compost. The difference between the bacteria that are in vermicompost is quite different than thermal compost. Mm-hmm. And so we need to be doing experiments on cannabis plants, since this is a cannabis show. Which one's better? Which bacteria, or is it a mix of the two? Because you want to get the right bacteria for your plant. So there's a great research project.
I feel like it's way too complicated, because when you mentioned just vermicompost, my immediate thought is, how long did the worms process the material? What species of worms did you use? What were the temperatures at? What was the worms fed? It could go on and on.
It could go on and on, but when it ends up at when it's gone through the worm 5 times, generally it has one set of bacteria in it, no matter what you start with. And same thing with compost. Not same thing, but I mean the compost, the base, again, I guess we ought to always go back to the base is the same for vermicompost, but different than the base for regular thermal compost.
Yeah, and I'd say the variability we see in compost commercially is, well, most of it's not even compost, but that's right. Much higher than in vermicompost. Exactly. So.
Oh yeah, but again, it's very different families of bacteria, very different, and so as a result, you get different things. And that that's an interesting thing that I had trouble with. We don't study bacteria, we study dinosaurs. And so if I was to write a book on dinosaurs, I could put in scientific names for a lot of different dinosaurs. And because a lot of the readers have been learning about dinosaurs from the time they were teething with their plush toys, all the way through, elementary school, they know dinosaurs, but we don't know bacteria. And the names are not easy. We don't have a common name for every bacteria. We have it for some, but not for everyone. Mm-hmm. And so, people shouldn't freak out by the fact that I list the names. You don't even have to pronounce them or try to pronounce them. By the end of the book, you'll know some of them. But I wanted there to be a place where they were listed, because we already have the database of bacteria. What we don't have is a home instrument that tells us what the RNA or DNA is for that particular bacteria that we can match with that database to tell us what that bacteria is. But we're getting there. And I think in the next 5 or 10 years, we're going to have that. We're going to be able to point the phone at a water down sample of soil, and it's going to tell us all of the bacteria that are in that soil. I think it's coming. I know we've got the microbiometer where we can tell biomass changes using our cell phone. It just makes so much sense for this to happen as well, and you can see a need for it in a really important way. So it's coming.
That I would be excited for, where we get a little more differentiation. Yeah, I could see that being a very useful tool.
Well, I'm not sure. We already have it. We can tell gram positive from gram negative, but we don't. As gardeners, we don't understand what the difference is. But again, all of these things are going to become clear and important as more and more people study this stuff, and as it becomes clear that using chemicals is creating a bad thing for the global warming, and there's just so many reasons for studying this stuff. It's just there. And they weren't there. They were there, but we didn't realize they were there back in 2006.
Yeah, and I just want to mention, as I'm looking through your book here, there's a lot more in there in regards to bacteria than just the rhizophagy cycle. Like you talk about how do bacteria communicate, how do they move, how how do they eat, and so you get to learn some of those things as well. How fast do they reproduce?
I do the same thing with the fungi book, because you've got to understand what a fungi is or what a bacteria is before you can understand what a rhizo or an endophytic bacteria is. And what I try to do is give just the amount of stuff you need to put the story together at the right. It should have been section 1 bacteria, section 2 rhizophagy, endophytes. Now that you know bacteria is what it is, because that's what it is. But you have to know a little chemistry here as well. How do bacteria talk to themselves? How do they? It's just that they are fascinating creatures, and we need to stop studying dinosaurs. We need to start studying microbes. And I say it all the time. I ought to start a foundation. I ought to have a group that's anti-dinosaur. So that we stop wasting our time on something that's useless and get into something that's useful.
Well, they do have microbe plush stuffed animals. I don't know if you've seen those.
Yeah, they do. They're beginning to get them, but
they do exist.
They're not. They're beginning to get a couple of books too. There's a couple of even soil food web books. So we're getting there, but we're not quite there yet. It's not taught, I don't think, in schools the way dinosaurs.
Now, one thing I want to touch on, because you bring up these commercial applications and commercially available products, and I think people get excited for that, as rightly they should. I've seen research showing the viability of some of these products. There's the regulation, I would say, on these sorts of things is all over the map. So it's not very well regulated because it's a new industry. So you could be buying something that's not what the label says, and I unfortunately see this all the time.
That's right.
So I just want to warn folks on the flip side of that, though. I see cannabis growers especially experiment with nutrients all the time. And I don't know why we don't consider doing the same with microbes. Set up an experiment in your environment with a control and see how your plants respond to adding one thing, in this case, a particular microbial inoculant. And I think that's a good way to approach it, because there is so much we don't know, and there are so many interactions going on. I think we really have to look at it in a case-by-case basis.
Oh, absolutely. I mean, I think there's a base that you want. I tell my clients there's a base for living soil. You want to have a good living soil, but then you want to tweak it, and this is one of the ways you can do it. And there are some products that we know are inherently unreliable. I don't mean they're bad; they're just unreliable, and you've got to experiment yourself to make sure you get the right one. I mean, mycorrhizal fungi is one of them. Some of them don't work, some of them do work. You gotta figure it out. You gotta figure out how to store them, how to take care of them, et cetera, et cetera. It gets a little bit more difficult with bacteria, because there's one kind, Bacillus. They form a dormant state that you can, it has a long shelf life, but most bacteria don't. And so how do you know whether you're buying it alive? It's, so you've got to be very, very careful. And you got to make sure that you're experimenting yourself. You can't take someone else's word for it unless you're awfully good friends with that person.
Yeah, I've unfortunately tried a lot of these sorts of products over the years.
Right.
Not seeing quite the results I want, but others I've seen amazing results. So.
Well, that's right, and that's why I like that microbiometer. I don't know. We keep coming back to it, is because you can test yourself. You can see whether it's working. Are you increasing your biomass in your soil? That's almost as good as, does the plant look better? You want to know. So you take a before, you take a couple of afters, or during, and you track. Information's power.
Yeah, I struggle with that one a little bit, just because I would think if you had a plant disease, it would increase your biomass as well, and that's where I'd love to know, like, individual species.
But you would know that. I've been asked that before. Yeah, but you would know that. You would see the plant being sick. But if you add, I won't mention a product, but if you add a well-known product that contains phosphorus and bacteria, you can take a test and know whether it's working or not. Obviously, if your plant's sick, you can take all the tests you want. It's still a sick plant. So I don't buy that argument.
That's fair. It's an interesting angle on. I hadn't really approached it from, because for me, testing biology is, whether that's sending it into a lab or the biometer, I guess I'm a little less interested in it than getting a mineral test, because a mineral test allows me to make, like, quantifiable decisions, actionable decisions about my garden. Whereas I feel like on the microbe side, I'm going to do all those things already, because, like you, I'm an organic guy. I want to take care of my soil. I know it's a little minor point of difference that we've had for a while. We've talked about this before, but but if people say to me, does this product team with microbes? I can tell them. Yes. I couldn't do it before I had the ability to measure it. And I think, you want that mineral test as well. Obviously, you got to have it, but you don't need to take that mineral test every 3 weeks. You can take a biometer test every 3 weeks. You know what I mean? So it's all information. You want all the information you can possibly get, and you want to know the pH, which is why it's been added to the. You want to know fungal, bacterial. There's lots of things you want to know, and so you try to find it out the best way you can. But if your plant isn't doing well, it doesn't matter. How do you account for the changes in microbial growth just based around hydrology? The water, the moisture levels in the soil.
Well, if your plant's not getting proper water, it's not going to grow well. But it's difficult. You can't. But by the same token, you've got to just be aware. So you have to be aware of seasonal. So biomass in fall is not the same as biomass in spring. Very different. The changes. And so if you're applying something in the spring to a plant versus fall, the biomass difference is going to be, or change or measurement number, it's going to be different. You just have to be aware of it. Nothing's perfect.
No, for sure. I was just thinking I'd want to control for that variable as much as possible. Like maybe I test the day after watering every time, so that I know that, because I've seen that your microbial communities will change dramatically. I mean, we know that bacteria can reproduce in 20 minutes, so you can have explosions quite quickly. So as ways to sort of level that out. I don't know. It's just
think of a compost tea, oh my God, the bacteria bloom you get, could be phenomenal.
It's huge.
Yeah. And again, what a wonderful opportunity to try to be able to identify the kinds of bacteria in various teas, and what they might do to to a plant, not as opposed to fertilizer type. In other words, not as opposed to creating the soil mediated bacteria, but to see whether there are any endophytic that end up. There's some implications here. And research that can be done, I'm sure. So
you brought up compost tea. That's where I started. That's kind of where you started.
Yeah.
I was having,
we all started with Dr. Elaine. She's the guru.
So I want to ask you a question, hear your thoughts on this. So
yeah.
I had a recent discussion with someone who posted an article on compost tea that showed basically overall good disease suppression in a study. And my sort of initial take is like, hey, that's great, but I don't think we can draw any real conclusions because every batch of compost tea is going to be different. The batch I make today, the batch you make, the batch I make 5 minutes after the first batch I make is going to be different. So because of that variability, I don't feel like I can really draw any major conclusions around other than the fact that we know that we're adding aerobic bacteria if we're making it correctly.
And some humic and maybe fulvics.
And some nitrogen. Yeah. But
yeah.
we have, Linda Chalker Scott, who's very anti-compost tea. I'm not saying anything that she would disagree with right there. She's kind of built a lot of her career around it. And part of the thing is because she cites a lot of research showing that compost tea doesn't work, essentially. And, we could poke holes in that research just as easily as we could, I feel like, poke holes around the research this gentleman was showing with disease suppression. But that being said, to me, the issue is not so much the research itself, but it's the inherent variability of these microbes because it's happening on a scale that's so small that we don't understand, that we can't control just because we're at such a macro level. I think it's one of those things that we really can't study. What do you think about that?
Yeah, I mean, that's always been the problem with compost tea is the variability of the compost. And not just the variability of the compost, but the way the tea was made, because they're all made differently. People put in different amounts, different brewers, different, you've got different temperatures, blah, 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 there's all there. And it's always been a problem. But I think the reason why, certainly from my perspective, I was initially excited about compost tea, and I'm still excited about compost tea. I don't use it very much anymore because I've gotten the benefits that I think I got. I think they came from the compost tea. Again, like you, it's very hard to prove. But I had a backyard that was so hard pan, and I never saw a bird there. There wasn't a worm anywhere. And today, as a result of applying compost tea, holy crow! I mean, the difference is literally night and day. And I attribute it to the application of compost tea. I've grown cilantro with and without compost tea in the same soils, and I've gotten in non-soil soils, perlite type stuff or vermiculite, and I've gotten differences better with the compost tea than not compost tea. Can I fully explain it? No, I can't. And there is a dearth of studies that say this is the greatest stuff since sliced bread. But again, as you say, a lot of it's got to do with the variability, and a lot of it you could pick holes through the studies. I mean, comparing compost tea in plants where you're using compost, that's giving the plant what the compost tea is. I mean, why is that a good study? So yeah, I've got the same questions you've got.
Yeah, I think what I'm looking for in compost tea is I'm applying a consortium. I'm trying to increase my nutrient cycling, that farmer model that you talk about. And maybe I'm getting some more of these endophytes in there too. But I'm certainly getting, an increase in bacteria and fungal biomass. We know that. And you can throw it under a microscope and literally see the nutrient cycling happen with the protozoa running around and the ciliates eating the bacteria.
Right.
And you can change the dominance of all of that. I know people talk about this by just brewing longer or adding certain food sources. So it can change dramatically. A tea at 12 hours is very different than a tea at 24 hours, even with all other variables the same. So this idea of trying to study it to me is just it almost feels like a fool's errand to a certain extent.
Well, I think sometimes it's an attempt to embarrass Dr. Elaine, frankly, but I wish people would stop doing that. But again, it's the variability is there. Many of us have had some very interesting results using compost tea, but some of us maybe didn't appreciate back then. Dumping compost tea onto a plant, depending on how you do it, can apply tremendous numbers of microbes, or it can be like putting a dropper full of compost tea into an ocean. What impact does it have? So it also depends on the receiving area as well as the compost tea itself. Yeah, it's fraught with bristling conundrums.
Yeah, I agree. I think at the end of the day, to me, everyone needs to just try their own, try it themselves, see how it works in their environment. And I think for the majority of people, it's gonna, if you're making it correctly, using good inputs, you're gonna see a positive benefit. But I don't ever lead with that assumption, or I would never say it's going to cure your fusarium or keep you from getting powdery mildew or some of the other things that I've heard around it.
Mm-hmm.
So,
I agree. I agree. You and I are not that far off.
It's funny how we all kind of fall into our little spots as we grow, and it changes. And I think that's what's exciting about it. And adding this book, Teaming with Bacteria, just brings a whole nother layer into this that's I think really exciting. So I appreciate you for writing this book.
Right. And it also explains some of the reasons why things don't work. Who knew that you had to have the right kind of bacteria to get the fixation that you want? Who knew? It's now we're getting a little better understanding. So there'll be many other books on this subject coming, I'm sure. And we'll know we're successful. We know we're successful with regard to soil food web stuff because it's what people study now when they go to college. They study the soil food web when they study soil science, and we'll know we're successful when the rhizophagy cycle is part of that study. And when we see it in articles and et cetera, et cetera. It's fascinating. I don't know how much time you want to waste on this stuff, but this year there's been a proliferation of articles about you don't need to rake your leaves in the fall. Now, to me, that's soil food web science in practical application in everyday life. You don't need to rake your leaves because the microbes will break them down, because they're valuable to feed your lawns, to feed your gardens, and you don't need to, even better, you don't need to use that polluting blower, et cetera, et cetera. This is practical application of soil food web stuff, and I love it. I love seeing it. And if you haven't seen those articles, you'll now be aware of them. And I guarantee you, in the next week or so, you'll see an Associated Press article or something, some clickbait article about don't rake your leaves. Here's why, and it's soil food web. Wow, I love it. A couple of years ago, pre-pandemic, when Apple came out with the I think it was the Apple Pencil. The iPad that they were using had a soil food web diagram on it. Wow! So this is really kind of fun. It's just fun to watch it all become real. We know it's real. We've been thanking Dr. Elaine and now Dr. James White, but now everybody gets to do it. It's really... It's very cool.
Yeah, the leaf things make sense to me. You're creating a natural layer of mulch. Every time the plant drops that leaves and you remove it, you're removing nutrients and organic matter from the environment that, unless you're replacing it, it's not a closed loop system. That being said,
right, it's a law of return. You're violating the law of return.
The one place I wouldn't want to do that is in controlled environment agriculture. So if we're talking to cannabis growers indoors, I would still suggest removing your leaves from the environment, from a disease perspective. But again, very different.
But I would use them. But I would use. Yeah, it is very different. But I would use them in the compost pile, and then use that compost in growing that cannabis.
Sure, you could absolutely
sure
compost it, thermally compost it. Absolutely, yeah, I don't have a problem with that.
Yeah, that's what I would do. And I think you can also vermicompost it as well if it turns out that vermicompost is better for your particular strain of cannabis. And it may be that vermicompost is better for all cannabis. I don't know. We need to do
that. Wouldn't remove the disease though risk, right?
Oh, wait a minute. You don't? Oh, yeah, I think it does. What doesn't remove the disease risk to me is anaerobic. A lot of the anaerobic mixtures can potentially have the diseases in it. But yeah, you put it through a worm, I'm pretty sure it doesn't have the diseases anymore.
I haven't heard that. I'll have to do some more research on that one.
Yeah, we're gonna, folks, look it up before you take my word for anything. But I've been using vermicompost on food products and cannabis for years.
Yeah, you probably had good starting inputs, though. I feel like I would be nervous. That's all.
Well, some of the poisons that I cook with. Are you kidding me?
Well.
Yeah, no.
I just want to say, Jeff, it's always a pleasure. I look forward to hopefully seeing you face to face here in the not too distant future.
Yeah, we should definitely. This post-COVID stuff, we need to start having these, these confabs again where we can get together. And of course, the Cannabis Science is available, and every year. And those are good conferences, and where we can kick some of this stuff around and stimulate other people to do more research to improve upon what's already been discovered.
Totally agree. Well, thank you again so much for your time today. For this book, I'll have links on the podcast page for folks that want to check it out. And I hope people will go out and read your book and start doing their own experimenting and research.
Sure. Right. And if they want to contact me, I'm a transparency, jeff@gardiner.com, particularly interested in hearing about your research. But if you got questions to ask, I don't answer immediately always, but go ahead and, I shouldn't say this on Tad's show because you have so many listeners, but we'll try to answer the questions. And if I don't know, I'll tell you.
All right. Thanks, Jeff. Well, have a good rest of the day, and we'll talk soon.
Oh, you bet. And say hello to the family.
That was Jeff Lowenfels, and you are listening to the Cannabis Cultivation and Science podcast. If you like the podcast, please leave us a rating and review and give us a follow on Instagram. If you want to support the podcast, check out the wider range of products we offer on our website, ranging from soils, amendments, beneficial insects, sticky cards, soil test analysis, and consulting. You can also sign up for our newsletter on our website homepage to stay up to date on the latest research and information. Thanks for listening.